Good morning. My name is Lynn Penberthy. I'm with the National Cancer Institute, and I'm here this morning to talk to you about cancer surveillance as an example of the role of federal agencies in advancing the use and progress in EHRs. The objectives that I'd like to cover today are to help you to understand the challenges in using EHR data specifically for cancer surveillance, with examples that include automated extraction using deep learning and NLP across heterogeneous healthcare providers, and source data linkages with use cases of pharmacy data and genomic testing. Using EHR for cancer surveillance is slightly different than applications with which you might be familiar, because central cancer registries have the legal authority to access health records under state public health reporting laws. As you know, most relevant information from EHRs consists of unstructured text. And this is an issue because the current process for surveillance is basically manual review and abstraction of data. And unfortunately, EHRs do not consistently capture information that's necessary for reporting on cancer patients because they're seen at multiple different facilities and treatment centers. Further, the process is manual, and so it's not sustainable for the increasing volume of data that's required to adequately capture cancer surveillance information, such as longitudinal treatment detail and genomic characterization of the tumors. So the solutions that we have focused on are targeting EHR components for automated data extraction and performing data linkages with source organizations that include external partners. Focusing initially on targeted EHR components, we have spent a lot of effort focusing on consistent and relevant EHR components that are useful for cancer surveillance, which include pathology reports and radiology reports, because they contain key data on tumor characterization and status, including site histology, laterality, behavior, and recurrence. There are challenges in using these path reports because there's inconsistent content from unstructured text that's received from the thousands of pathologists who record their path reports. In addition, there are more than 500 histology categories and 100 cancer site classifications, which really impact the training data that we have to enable robust algorithm development, specifically for rare tumors and histologies, which can represent class imbalance challenges for deep learning. In addition, the registries require very high accuracy, greater than 97%, across all of the data elements that are being extracted through automation. We're now working with the Department of Energy in a collaboration to develop algorithms to extract key structured data from unstructured pathology reports, because we receive more than 500 types of pathology reports from path labs across the US. There are 4 to 5 million path reports that are received annually in real time at the registries. So we see that there's real value of automation. As I mentioned earlier, the data are currently manually reviewed and annotated or abstracted. And so automation would significantly reduce the labor, because there's an estimated more than 60,000 person hours per year that are used just for the screening and extraction process. In addition, SEER is moving towards real-time reporting of incidence data, and it's not feasible to do this with the manual extraction process, which is delayed. So the solutions that we've been developing use existing gold standard manually extracted data from 7 registries to train the algorithms and to develop APIs for processing the unstructured path reports. We're doing iterative testing and development with review of mismatches for retraining of the algorithm. And we've developed uncertainty quantification processes that helps us to identify the path reports where there's less than 97% accuracy across the structured data that's extracted and sent for human review. The current status of this process is that we've implemented into the production workflow the API in 11 SEER registries, and this allows us to automatically process between 23 to 25% of all of the more than 4 million path reports that we receive annually. That is, there's no human review. And we've achieved a greater than 98% accuracy across the 4 data elements that we're focusing on: site, histology, laterality, and behavior. The API is 18,000 times faster than human, and this translates into savings of more than 11,500 labor hours for this process alone. We're also leveraging this workflow to iteratively improve the algorithm through feedback from the manual process and by doing a 10% random sample of the manual review for auto-coded reports. And we're using it to train personnel to increase the consistency and accuracy of those reports that are not auto-coded. Lastly, we're leveraging the API that we've developed for recurrent metastatic disease for path reports and using transfer learning to apply that to radiology reports. Next, I'd like to move on to data linkages, leveraging source data for surveillance. Linkages with source data, that is external partner organization, really offers the surveillance community an opportunity to maximize all matches for data that are generated by the source rather than through the EHR. And I'll give you an example of this for Oncotype DX, which was manually extracted from EHR data since 2012. And when we did the linkage with the company directly, that is the source data, we found that we were missing about 40% of all tests. These linkages are done under surveillance reporting regulations, as I mentioned earlier. And I'd like to move on to two use cases for pharmacy data and genomic test panels. SEER registries receive currently all anti-neoplastic drugs that are provided by Walgreens, CVS, and Rite Aid because pharmacists are covered under state public health reporting regulations. This really provides important information on treatment that's not routinely available to hospital registries through their EHRs. And this is important because we capture treatment for both the initial course of therapy as well as subsequent treatment of recurrent disease. And this gives us an opportunity through the longitudinal coverage to potentially identify patients who have recurrences. Here's an example of some of the SEER-linked pharmacy data from 2013 to 2020, 3 different types of agents. And I just wanted to quickly go through this to illustrate the magnitude, is that there were more than 20,000 patients who had TKIs during this period, and almost 2,000 patients who had PARP inhibitors, and about 4,300 patients who had a CDK inhibitor. And so I think you have a sense of the magnitude and the importance of this information. There are, of course, challenges that largely related to prescription coverage by insurers in the US because it's very heterogeneous and inconsistent over time. In fact, certain anti-neoplastic drugs are only provided by designated providers, such as CVS, for example, or Cardinal Health, and that can change annually. And so this represents a challenge for how we release the data in SEER because there's a risk of bias if we don't have complete coverage. And so the solutions that we've come up with, which I think may be relevant for other uses of EHR or similar data, in that we release the data under carefully controlled circumstances with review of proposed analyses. And we're currently performing sandbox testing of the linked data to help us understand where there are biases. And we're comparing with more comprehensive data sources such as SEER Medicare Part D to help us identify those gaps and how we might fill them. And of course, we're continuing to increase coverage by working with additional partners such as mail order pharmacies and pharmacy benefit managers. The second use case is genomic data. And unlike pharmacy data, the linkages with selected genomic path labs provide comprehensive information on all patients receiving the test. Examples include Oncotype DX, for breast, Oncotype and Decipher multigene panels for prostate, and Castle multigene panel for melanoma. The challenges here are a little bit different and really relate to assuring appropriate matches because patients move over time and they may have a genomic test at some time subsequent to the initial diagnosis. And so matching based on name, date of birth, and address may not reflect an accurate match because of the timing. And so the solution that we've come up with is SEER registries now link with LexisNexis to obtain longitudinal residential histories that can permit adjudication of questionable matches, allow us to do manual review of those uncertain matches, and to increase the match rate significantly. So in conclusion, using currently available EHR data for cancer surveillance remains challenging, but specifically targeting EHR components can provide significant value. And in understanding what components of a patient's healthcare EHRs actually contain is really critical to optimizing that value. So for example, are you looking at community oncology practice EHRs or freestanding radiology centers or radiation oncology centers? And then lastly, of course, as you all know, this is not a fast or easy process. Thank you very much for your attention.